Next, we'll have Philippe Jawinski from Humboldt University in Berlin tell us about some cool results on GWAS of EEG oscillations.
Yeah, so many thanks for having me. I'm super grateful having the opportunity to present you with some of our most recent GWAS results of the Enigma EEG working group. So in my talk, I will delve into the genetics of EEG oscillations and provide you with some novel biological insights into the intricate links between brain structure, function, and behavior. I have nothing to disclose. So just to make sure that we're all on the same page, the EEG oscillations have been discovered in 1924 by Hans Berger. So this year we can celebrate the 100th anniversary of the discovery of EEG oscillations. They play a crucial role in neuronal information processing and have extensively been studied as markers of as human behavior and psychopathology. Importantly, twin studies, they have shown that they have a strong genetic component. But unfortunately, the individual genetic variations that contribute to the strong heritability, they remain largely uncertain. And this is basically due to a lack of power that we have for these kinds of analysis for genome-wide association studies. So in Enigma EEG, we joined forces of 9 different cohorts with an aggregated sample size of about 14,400 individuals. All of the different cohorts, they had resting state EEG data available, resting state EEG recordings of about 3 to 5 minutes. They also had genetics data available, of course, and we were interested in 7 super basic resting state EEG variables. We were interested into alpha power, beta power, delta, theta broadband power at the vertex site, as well as alpha power and peak frequency at occipital leads. So we measured about 10.5 million genetic variations, mainly SNPs, but also some kind of short insertions and deletions. And the total sample size, again, was 14,400 individuals. The main objectives were to discover individual loci associated with the EEG oscillations. But also we wanted to explore the genetic relationships, the overlap with other complex traits like mental illness and brain structure. For reasons of reproducibility and also for reasons of transparency and reproducibility and for reasons of collaborational work, we uploaded all of our analysis scripts to a GitHub repository, which will be made publicly available along with the preprint. All right, so after we've run the GWAS meta-analysis across the 14,400 individuals for the 7 EEG variables, we were first interested in the heritability, which was estimated using LD score regression. The SNP-based heritability ranged between 14 to 27%, which should be understood as a lower bound estimate of the total heritability because we're not considering structural variations like C and Vs here. We are mainly focusing on SNPs. So the SNP-based heritability ranged between 14 to 27%. And to put this into perspectives, we compared this against MRI estimates or heritability estimates derived from the MRI from the UK Biobank cohort. There are publicly available data of about 4,000 MRI-derived phenotypes, brain MRI phenotypes, which were measured in about 32,000 UK Biobank individuals. And what we see is that with the SNP-based heritability of about 14 to 27%, the EEG variables, their heritabilities, they are larger than those of functional MRI traits. They are average to above average when compared to structural MRI traits, and they are average to below average when compared to the heritability estimates of diffusion MRI traits. So the bottom line here is we have a strong genetic component comparable to the heritability estimates from structural MRI traits. What you can see also on the slide is something else that you can do with the LD score regression. You can check the genetic correlations between the different EEG variables. This is what we did. What we see there is a strong cluster of strong positive correlations between the power variables, alpha power, beta power, delta. They have strong positive correlations, and we also see negative correlations between power and alpha peak frequency. And these genetic correlations, they well reflect the phenotypic correlations. So this can also be understood as a sort of a sanity check. Yes, but bottom line, we have a strong genetic component, and this is a good position to identify individual loci. And that's what we did next. So on the left side of the slide, you can see the different Manhattan plots, 7 Manhattan plots for the different EEG variables. So there are about 10.5 million dots in each of these plots, reflecting the associations of single genetic variants that we associated with the respective phenotype. Across the 7 GWASs, we identified 2 genome-wide significant loci. Both of them were associated with alpha peak frequency. The more interesting one of them is an intronic region of the FANCA gene. It is a multi-tissue expression quantitative trait locus, so it has previously been associated with the expression of certain genes. It has also been associated with the risk for schizophrenia, which is quite interesting, I think, and a couple of other traits. So we got 2 genome-wide significant loci, which actually is, so we hope to discover more of them, but in the end we had 2 genome-wide significant loci. And this basically perfectly corresponds to what we see in other highly heritable traits, where the first genome-wide significant discoveries are found in samples of about 10,000 to 20,000 individuals. There are some other techniques that can be used, methods like the pleiotropy-informed conditional false discovery rate approach, which allows to boost the statistical power of a GWAS. It basically borrows the power of larger GWASs, for example, the GWASs of the Psychiatric Genomics Consortium, which have been conducted in hundreds of thousands of individuals. And we now can condition our results on the evidence that has previously been derived for the variants that we also assessed, but in larger GWASs. And by applying this technique using GWASs, for example, for major depression, for bipolar disorder, for schizophrenia, and so on and so forth, we discovered 34 loci, which likely contribute to the heritability of the EEG oscillations. This is a map of those 34 loci. So at the top, you can see a merged Manhattan plot across the 7 EEG variables with the 2 genome-wide significant loci, which originally were found for alpha peak frequency. And on the bottom of this Miami plot, you can see the 34 identified loci. 27 of them have a nearest gene that is protein coding, 24 tag known G-tags EQTLs, so they have previously been associated with the expression of certain genes, and 11 loci, they were associated with variants that are also reported in the GWAS catalog. So these variants have been linked, for example, with psychiatric traits, unsurprisingly, but also with cognitive ability, with alcohol consumption, executive function, and neuroticism. So there appears to be a pleiotropy with other complex traits, which brings me to our next analysis, the genetic correlation analysis with ALD score regression. So on the left side of the slide, we've run genetic correlations for our 7 EEG variables and a bunch of selected traits from the psychiatric domain, from substance use domain. We also had some neurological variables, personality variables, sleep variables, and so on and so forth. We found the most compelling support for a genetic correlation with generalized epilepsy, which makes sense, I think, because generalized epilepsy is closely related to electroencephalic activity, of course. The association is in the way that genetic variants that contribute to stronger power in the EEG are also variants that contribute to higher vulnerability towards generalized epilepsy. To our knowledge, this is the first study showing genetic correlations between brain MRI variables and EEG oscillations. The strongest associations were found with global markers like total surface area, but also with cerebral white matter volume. The top regional associations, they implicated the superior frontal, the orbitofrontal, and the precuneus surface area. We also, in a more agnostic approach, we also run genetic correlations with 1,000 traits of the UK Biobank cohort, but these associations, shown at the right bottom, none of them reached significance after correcting for the 1,000 tests that we've run. All right. And last but not least, we were also interested in the degree of polygenicity of these EEG oscillation traits. And there's a method that is called a genetic effect size distribution analysis, which allows to calculate or estimate the number of underlying susceptibility variants that contribute to the heritability. And for example, for broadband power, this number has been estimated at about 5,500 variants that contribute to the heritability, which is comparatively low when compared to other traits. For example, human height has been estimated at about 12,500 variants. neuroticism has been estimated at about 16,000. And taking a look at the plots, left side, we see the distribution of genetic effect sizes. For broadband power, we have these thicker tails indicating that there's a relatively large number of variants or greater number of variants with strong effect sizes when compared to height and neuroticism. And it also allows to make some prospective statements, predictions about what happens if we have larger sample sizes. So with increasing sample sizes, we expect a relatively rapid increase in the genome-wide significant discoveries and also a rapid increase in the genetic variance that is explained by those identified genome-wide significant discoveries relative to the other complex traits. All right, let me summarize. So I just showed you what is to our knowledge the largest GWAS on EEG resting state variables to date. In accordance with twin studies, we find a substantial genetic component for the EEG oscillations. We identified 2 genome-wide significant variants, both of them for alpha peak frequency. One of them is schizophrenia risk variant. We identified 34 loci via pleiotropy-informed analysis with psychiatric traits. Using genetic correlations, we also find evidence for genetic overlap with brain structure variables, generalized epilepsy, and also indications for an overlap with neuroticism and loneliness. And using genetic effect size distribution analysis, we also find that EEG oscillations, they are complex traits, but when compared to other complex traits, you may say that they have a relatively low degree of polygenicity. All right, this kind of research would not be possible without the contributions of many researchers. I would like to thank all the contributors, all the members of the Enigma EEG working group for their valuable contributions, and I would like to thank you for your attention, and I'm happy to take questions. Thanks.
Questions from the audience?
Thank you for the great talk. I was wondering, in light of the high genetic correlation that you reported, have you tried any multivariate GWAS to try to maybe boost power?
Which high genetic correlations do you mean? Genetic correlations between which variables?
Between the different EEG measures?
Okay, yeah, so the different EEG variables, they indeed show very high correlations. We didn't do like partial correlations so far. I guess this would be quite interesting also to partial out the effects of the other EEG variables to see what are the associations with those variables. We haven't done this for now, but yeah, this definitely is an interesting idea. Thanks.
Thank you so much for your great talk. I have a question about, again, genetic correlation of like EEG activities and surface area. But I was wondering, like, what is the underlying mechanism of, of that correlation between surface area and EEG activities? Because you probably didn't find any correlation between, for example, EEG activities and cortical thickness.
No, we didn't find significant correlations with cortical thickness. We found them with volume and with surface area, in the sense that variants, so these genetic correlations, they were negative in the sense that variants that contribute to stronger power of the EEG, they contribute to lower surface area. And we were, in the poster session, we were discussing about this, and the idea is maybe that if you have a lower surface area, that the signal from within the brain is like more condensed if you have lower surface area, which makes it like larger if you measure it. But I'm not 100% sure whether this really is the answer, but yeah, that's a possible answer.
Thank you very much.
Thanks.
We might have time for one more question. Okay.
So again, to boost power, did you try any kind of polygenic risk score to construct something like that, or did it work, or something?
We didn't do it yet. We try to. You mean? Okay, so there are two things that you could do. You could use the GWAS meta-analysis results now and calculate polygenic scores in other samples and correlate them with other variables. That's something that we could do, for example, in the UK Biobank cohort. This is also something that is on my to-do list, actually, but I'm not 100% sure whether I will really do it, because I'm not sure. I first have to do some statistical power analysis, whether this is really reasonable. And yeah, we've been thinking about doing this in one of the sub-cohorts, just like for the meta-analysis, leaving out the data of one of the sub-cohorts, then calculate polygenic scores, for example, in one of the larger samples, because it's always a little bit difficult if you have these collaborative projects to tell everyone to do the polygenic score analysis, because then it often takes months to get the results completed. But yeah, we haven't done this so far, but it's something that we may do in one of the subsamples, yeah, the larger ones. And would you calculate the polygenic score, for the EEG variables and correlate them with other variables? Or would you calculate polygenic score, for example, for schizophrenia and see whether the polygenic score for schizophrenia is associated with the EEG variables? The second one, okay, yeah. Yeah, this, yeah, we could also do this. Yeah. Yeah, we haven't done this so far, but yeah, this is a very good idea. Maybe for another project, maybe we'll take it into this one, but yeah, maybe we'll also start a new manuscript.
I'm trying to do something similar, so this is why.
All right, okay, cool, thanks.
Thanks so much, Philippe.